Lobster Stock Enhancement. Clawed lobsters of the Homarus species are distributed on both sides of the North Atlantic and in the Mediterranean. On Atlantic coast, they support significant commercial fisheries and provide valuable employment in coastal communities. Due to increasing demand, stocks are subjected to heavy fishing pressures and are close to natural sustainable yield, and Mediterranean stocks are generally at or approaching sub-viable levels. Despite attractive market prices, due to high grow-out costs, intensive cultivation of lobsters to market size has not proved economically viable. Ranching appears to be a more viable option, and various programs have been undertaken in Europe and North America to enhance catch levels by release of hatchery-produced juveniles into the wild. For example, Lee and Wickens in 1992 report commercial catches comprising of 4% to 50% of experimentally released lobsters in a specific fishery. These programs may also be of importance in developing new fisheries on artificial substrates, restocking sub-viable fisheries in the Mediterranean, or those depleted by a cataclysmic event. Lobster interests have identified stock enhancement or ranching programs as the most promising method for securing the long-term future and development of the industry. The fishing for lobster provides an essential form of income to many rural communities around the Irish coast. There have been large fluctuations in landings over the years, and this has caused instability in the industry. There is also a high demand and high unit price for the European lobster, Homarus gammarus, which has attracted much attention for aquaculture and stock enhancement initiatives. It is for these reasons that a lobster restocking program has been established at the Shellfish Research Laboratory, part of University College Galway, in Carna. This work is being carried out in conjunction with Céidigh Cho. Céidigh is a mariculture research and development company funded by Údarás na Gaeltachta, the board representing the Irish-speaking areas of Ireland. This project was initiated in 1992 to investigate techniques involved in the culture and release of stage 4. In November 1992, at the Irish-American Technical Exchange on the aquaculture of abalone sea urchins, lobsters and kelp, Brian F. Beale spoke about lobsters, Homarus americanus culture in Maine, a community-based, fisherman-sponsored public stock enhancement program. He described the methods that he and his colleagues had developed and used at the main fishing village of Cutler to produce up to 150,000 stage 4 and 5 animals each year, with the typical survival rates through their larval period of 30 to 50%. After this paper and a subsequent visit to the Shellfish Research Laboratory by Brian Beale, much of the methodology used at the Cutler hatchery was adopted. 4 species of lobsters are found in Irish maritime waters. 2 are clawed lobsters, Homarus gammarus, the European or the blue lobster, the subject of this video, and Nephrops norvegicus, the Norwegian lobster or Dublin Bay prawn, usually sold as scampi. There are also 2 spiny lobsters, Palinurus elephas, the crawfish or cray, and Palinurus mauritanicus, the pink crawfish. Legislation. There are no regulations for the capture of spiny lobsters. However, minimum size limits apply to clawed lobsters as follows. Homarus gammarus, the European or blue lobster, has a minimum size of 85mm carapace length or 24 centimeters overall length. For Nephrops norvegicus, 25mm carapace length or 85mm overall length exists. A minimum mesh size of 70 millimeters, knot to knot, applies to the Nephrops trawl fishery. For all lobster species, there is no legislation to prohibit the taking of buried females, for an upper size limit, for escape holes for juveniles, to limit the number of traps or the type of fishing gear used, or to limit the season, although nature enforces this with weather conditions in most fisheries. Landings of lobsters, European lobster, Homerus gammarus, in Ireland over the last 10 years have varied from 281 tonnes to 516 tonnes per annum. The price has varied from a low of £3.76 or $5.41 per pound to a high of £5 per pound or $7.20. The broodstock, or buried females, are bought or exchanged from a local lobster merchant, Cleggan Lobster Fisheries Limited. The selection of the broodstock. Each buried female is carefully inspected, and those chosen are large females being taken in preference to smaller ones because of their superior fecundity, strong, active and intact animals, and only lobsters with large amounts of healthy eggs. Once the broodstock have been selected, they are weighed and carefully packed into a polystyrene fish box with their tails tucked underneath them to protect their eggs. They are covered with damp sacks or sponge and a polystyrene lid placed onto the box. It is important that they are kept cool and moist and still while being transported to the Shellfish Research Laboratory, a journey time of about 35 minutes. When the eggs on the lobster have changed to a reddish-brown colour, the females are brought into the hatchery. The eggs of the female lobster are bathed in a 1% solution of iodine and seawater that has been UV treated and filtered down to 1 micron. All but the mouthparts of the animals are submerged in the solution for up to 7 minutes. The bathing in the iodine solution is a procedure that is undertaken to remove as many external parasites and pathogens as possible. After being bathed in the betadine solution, the broodstock are rinsed in a water bath that has been filtered down to 1 micron. They are then placed into broodstock bins. There are 4 of these 500-litre bins, and these are filled with UV-treated seawater. Shelters made of PVC pipe are provided to reduce stress and fighting amongst the broodstock. The broodstock release their larvae at night. Because lobster larvae are cannibalistic upon hatching, they are collected from the 500-litre tank within 1 hour. Another reason for swift collection is that the female present may prey on these early-stage lobsters. Weight measurements are taken by dab drying the larvae in a net on tissue paper and then weighing them. The reason for weighing the larvae is in order to estimate their number. Once the larvae have been weighed, they're placed into a 70-litre hopper containing 2 species of algae. Artemia are commonly referred to as brine shrimp. Unlike other food organisms, for example rotifers and copepods, that have to be maintained in continuous culture, Artemia can be produced when required using inert dry cysts. Diverse groups of organisms have been successfully offered Artemia as a food source. The inert dry cysts are placed into a 1 litre beaker. To this is added chloros. The chloros removes the outside shell of the Artemia, and in doing so disinfects the Artemia cysts. The decapsulated Artemia are rinsed using fresh water and placed into a 3 litre beaker. To which is added 2.5 litres of UV treated seawater. The Artemia hatch after 12 hours. They are then added to the 70 litre hopper with the lobster larvae. Here we see 14-day-old lobster larvae that have reached stage 4. These lobsters are now ready to be taken outside and placed into their individual compartments. The lobsters are scooped out of the hopper and weighed. They are then placed into a 500 litre tank. The aeration to the 500 litre tank is turned off in order to separate stage 3 lobster larvae from stage 4s. The stage 4 larvae are transferred using a tea strainer into individual compartments. Adult Artemia, which have been cultured in a 500-litre bin being fed on an algae called Tetraselmis, are harvested by sieving. They are then placed into a water bottle and fed to the juvenile lobsters in their individual compartments. At the Shellfish Research Laboratory, we have a large range of young or juvenile lobsters being grown. These lobsters that are being fed are 1 year old, and they're being fed on green crab legs. When sufficient quantities of juvenile lobsters are available for release, they are tipped into a flow tray. The lobsters are scooped up using a net and placed onto wet tissue paper and a mesh. They are then evenly dispersed around the tray. The lobsters are covered with another layer of mesh and tissue paper, and the trays are placed into a polystyrene box. The polystyrene box is covered and sealed. There is a journey time to the release site of approximately 4 hours. The release site shown here is in Smerwick Harbour, County Kerry. All equipment is loaded onto the boat early in the morning. The boat trip to the release site takes approximately half an hour. The site at which the lobsters are to be released has been previously surveyed by a diver as to its suitability for releasing lobsters. Temperature, salinity readings are taken at each release site. A 30-meter hose is lowered over the side of the boat. This pipe is used for deploying the lobsters onto the seabed. A diver observes the release. The lobsters are taken from the boxes. And placed into 10-litre buckets. They are then poured down the 30-meter hose down to the seabed. The lobsters can be seen coming out of the pipe. We would like to acknowledge the support for the Lobster Stock Enhancement program shown in this video, provided by Údarás na Gaeltachta. The program is a spin-off from the Irish-American Aquaculture Initiative, a joint Sea Grant Irish venture started in 1989 and its subsequent meetings. Thanks are due to Dr. James McVeigh of the National Sea Grant and Professor Ed Monaghan and his team from Sea Grant Northeast for their efforts in developing their transatlantic cooperation. Dr. David Dow of the Lobster Institute facilitated. Special thanks are due to Professor Brian Beale of the University of Maine at Machias for his efforts in helping the program through its crucial formative stages. Together with others too numerous to mention, Moylotte.